Hello everyone, welcome to our channel Biology for All. In this video, I'm going to talk about the structure of DNA. DNA or deoxyribonucleic acid is made up of two polynucleotide chains. When we say polynucleotide chains, that means it's made up of many nucleotides. Nucleotides are the monomers that make up the polynucleotide, and it's a single unit. Let's see here the structure of one nucleotide. A nucleotide is made up of three distinct parts. The first part here is the phosphate group. Then it has a sugar, that is a pentose sugar, a 5-carbon sugar, and a nitrogenous base. Let's have a look at these three structures. A phosphate group has a phosphate with the three OH groups attached to it. The pentose sugar shows two forms, ribose and the deoxyribose form. So you have the structural formula here, and we can see that if we number the carbon atoms, then at carbon atom number 2, it's seen that there is an OH group in the ribose, whereas there is no OH in the deoxyribose. Ribose is present in the RNA, and deoxyribose is the sugar that is present in the DNA. The third component of the nucleotide is a nitrogenous base. Nitrogenous bases are heterocyclic compounds. Heterocyclic because they have nitrogen along with carbon in the ring. These are of two types, purines and pyrimidines. Purines are double ring structures, whereas pyrimidines are single ring structures. If we number the structure here, we are able to see that in purine, nitrogen is present in position 1, 3, 7 and 9, whereas in pyrimidine, it is in position 1 and 3. Purines are adenine and guanine, whereas pyrimidines are cytosine and thymine, uracil in case of RNA. So here, let us now have a look at the formation of a nucleotide. We have seen that the nucleotide contains a phosphate group, a pentose sugar, and a nitrogenous base. Now, what bonds connect these three? Let's have a look here. This is the structure of a deoxyribose sugar, and I'm drawing here in green a purine and a phosphate group here in red. Now, the bond formation happens between carbon atom 1 and the nitrogenous base and carbon atom 5 and the phosphate group. So, if we now draw a structure combining these showing the bonds, then the bond between the sugar and the purine or the nitrogenous base is the glycosidic bond, and that glycosidic bond is written as N-glycosidic bond because there is a nitrogen present. It forms a bond between carbon 1 and N9 in case of purine and carbon 1 and N1 in case of pyrimidine. The bond formed between the sugar and the phosphate is called as a phosphoester bond. So this makes up the structure of a nucleotide. A simple schematic representation can be given as follows. So when we are now drawing the structure of a DNA, then we draw a nucleotide in the following simple representation. So here I have three nucleotides together. Now a bond formation in order to form a polynucleotide chain happens between the phosphate group and the third carbon OH. So this bond now that is formed will be called as the phosphodiester bond between the two nucleotides. So here is one strand of the DNA molecule with three nucleotides, and again I'm drawing on the other side another strand with three nucleotides and showing the phosphodiester bonds. Now these two strands are connected together by weak hydrogen bonds. We say weak hydrogen bonds because these can be broken easily. The two strands that are connected by the hydrogen bonds run anti-parallel to each other. So what do we mean by anti-parallel? they run opposite to each other. If you look at the pentose sugar, it can be seen. Now, if you consider the strand on the left side, then we can see that on the top you have a phosphate which is free, and at the bottom there is an OH group that is free. So here at the bottom of the strand there is an OH group attached to the third carbon of the pentose sugar, which is the free end of the strand. So you can add nucleotides here. So this is called as the 3' end. The top end thus will be called as a 5' end. The other strand on the right side will have it in the opposite numbers. Now, the nitrogenous bases are in the form of complementary bases. So here we have A pairing with T, C pairing with G, A and T having 2 bonds, and C and G having 3 bonds. The sugar and the phosphate together forms what we call as a sugar phosphate backbone of the DNA molecule. So this is the structure of the DNA molecule.